What is the difference between planktonic and sessile bacteria? Imagine tiny creatures in a drop of water, some swimming freely while others are stuck to a surface. That's the core of what makes bacteria fascinating in the world of microscopic marvels. You might think all bacteria are just simple germs, but their lifestyles are surprisingly diverse. Today, we're exploring how bacteria live and thrive in very different ways. First up, planktonic bacteria are like tiny explorers floating around in water or other liquids. They are single cells that move by themselves, swimming or drifting with the flow. These bacteria are usually fast growers because they don't have to worry about sticking to anything. They spread out quickly to find new places to live. But being free-floating also makes them more vulnerable. If a medicine or disinfectant comes along, they can be easily swept away or killed because they lack a protective shield. Think of them as lone adventurers, always on the move, ready to colonize new environments. Now, on the flip side, sessile bacteria are the homebodies of the microbial world. Instead of wandering around, they attach themselves to surfaces like rocks, pipes, or even your teeth. Once they settle, they start building a community called a biofilm. This biofilm is like a slimy city made of bacteria and a sticky matrix of sugars, proteins, and DNA. This matrix acts like a fortress, protecting the bacteria from antibiotics, cleaning chemicals, and harsh conditions. Because they are in a community, sessile bacteria grow slower but work together, communicating through chemical signals. This teamwork makes them super resilient and hard to wipe out. Interestingly, bacteria can switch back and forth between these two states. When conditions change, sessile bacteria can break free from their biofilm and become planktonic again. This ability to change modes helps them spread and survive in different environments. Detached bacteria often show traits of both lifestyles, making them tricky to control, especially in hospitals or industrial settings. In bizarre biology experiments, scientists have seen biofilms form on strange surfaces like contact lenses, inside pipes, or even on ship hulls. These slimy bacterial communities can survive treatments that normally kill free-floating bacteria. That's why biofilms are often the culprits behind stubborn infections and equipment failures. They turn tiny bacteria into microscopic fortresses that resist destruction. Understanding the difference between planktonic and sessile bacteria is key for many fields. In medicine, it helps us fight infections better. In industry, it guides cleaning methods. And in biotechnology, it opens doors to harnessing bacteria for useful purposes like cleaning pollutants or producing biofuels. So, to sum it up, planktonic bacteria are the wandering, free-floating explorers, while sessile bacteria are the community-building, surface-attached survivors. Their ability to switch between these two modes makes bacteria some of the most adaptable and strange creatures in the microscopic universe.